All right, we kind of just reviewed this, but let's look at it again. Everything you do is metabolism. Everything you do is a chemical reaction. All the chemical reactions in your body, the sum of those, make up your metabolism. We typically divide metabolic reactions into two kinds: catabolic and anabolic. And we just talked about how catabolic reactions are what kind of reactions? Help me out here. Catabolic reactions do what? Again? They break things down. What's another word for reactions that break things down? Hydrolysis. They're going to usually give off energy, which means they're typically what kind of reactions? Exergonic. And since they're giving off energy, a lot of that's heat. What do they do to entropy? Increase it, right? They're releasing heat. Heat is entropy. Heat is disorder. So, well, let's move on to this next one first, and we'll come back to that. Anabolic reactions, on the other hand, are putting together. We call those synthesis reactions. What are two other words we use to talk about synthesis reactions? Condensation and dehydration. So we might say anabolic, we might say condensation, we might say dehydration, we might say synthesis. They all mean the same thing. They mean putting together, building up. Typically, it takes energy to make bonds. So those are going to be endergonic reactions. And those reactions are organizing things. They're taking in energy and increasing order, therefore decreasing entropy. Does that make sense? Another big idea that you need to make sure you understand is the idea of coupling. We know couple means to pair up, to put together. In biology, what two kinds of things always get coupled? Yeah, an endergonic reaction, before it can happen, it has to have energy. And the energy to make it happen comes from where? Exergonic reaction. And that exergonic reaction is almost always which one? What is your body's most important exergonic reaction? Yeah, and the word for that is? The breakdown of ATP, what's it more scientifically called? ATP, the breakdown of ATP. You just told me the word a second ago. Hydrolysis, okay? ATP hydrolysis. So ATP hydrolysis provides the energy for just about every endergonic reaction in your body, the activation energy, the energy needed to make it happen. That's ATP cycle diagram. It's very important that you understand that. ATP is what kind of macromolecule? Of the four groups we talked about over and over again. ATP is in which group? Nucleic acids. It's in the same group as what other two important things? DNA and RNA. It's built out of nucleotides. Each nucleotide is made out of a sugar in the middle, and that sugar is what sugar? Let me give you your picture. What's the sugar in ATP, guys? Ribose, same one that's in RNA. You need all five of those things. Over here, this part in orange, what's that? The nitrogenous base. There you go. Which is which nitrogenous base since it's ATP? Adenine. And it's got 3 phosphates. Now it's important to realize when ATP gets hydrolyzed, what gets broken typically? That's that bind right there. The bind between the second and third phosphates. That's what gets broken, and therefore that releases energy, heat. Typically, how does the energy get from ATP to where it needs to go? How does the energy get transferred from ATP to something else? This is something you got to understand. It's a process called phosphorylation. The phosphate gets broken off and it gets transferred to something else. Maybe an enzyme. maybe a protein. So the phosphate is broken off of ATP, and it's transferred to some enzyme or protein. Yep. And what does that phosphate carry with it? The energy. That's how the energy is transferred. It's a way of transferring energy from ATP to something else. Phosphorylation. Break off the phosphate, attach it to something else. That process of hydrolysis and phosphorylation provides the energy for just about every endergonic reaction in your body, whether it's making new proteins or making nerve impulses or moving your muscles or anything else. ATP hydrolysis is coupled with just about every endergonic reaction in your body. But when it's all done, what's left is ADP and P. We set to think of those like what? ADP and P are like dead batteries. Dead batteries, but capable of being recharged. And the process in your body that recharges your batteries, literally, is which process? Cell respiration. So that's what's going on over here, is cell respiration. And the energy to recharge your batteries comes from where? Food. While we eat. So the ATP cycle is huge. You got to understand that. You got to be able to explain it. That's essentially the same diagram that was on the question at the beginning of class today, right? It's essentially the same thing. Hey. You're looking for, she's back in that corner today. That's okay. All right. You just want to take a time out and teach about Gibbs free energy while you're in here? I know you love her while you're in here. Just let her teach today. We could let Jones teach. She doesn't know it. What's she saying about you?
We haven't done that yet.
I thought you were calling her names or something.
No, I was looking for old.
Okay.
Like this.
Gotcha. All right, guys. So we got the Gibbs free energy equation. We've already looked at it. You guys by now ought to know. You just ought to memorize the equation. We're going to call that delta G. What? What is delta G? Change in free energy. And the way I want you to think of free energy in this class, that's a pretty good definition up there up on the slide. It's the energy that makes things happen in your body. It's the energy that makes reactions get started. What other phrase do we often describe that by in biology?
Free energy?
Free energy, or what's the energy that makes reactions get started oftentimes called? Activation energy. We might use that as another word for this. Make sure you know that. Free energy is equal to the change in enthalpy, which is the change in the total amount of energy in the system, or in the body in this case, minus the temperature. That temperature's got to be in what unit? Got to be in Kelvin. Got to be the absolute temperature. And that temperature gets multiplied by the change in entropy. And another name for entropy is what? Chaos, disorder. Now, the numerical value of that's important, but maybe the more important thing for you to realize is what happens with the sign. If you get a negative delta G, you ought to know about 4 things that should tell you. Negative delta G, first of all, is telling you what is happening with the energy from the reaction. It's being given off. It's releasing energy, which means it's what kind of reaction if it's releasing energy. Exergonic. It also means it's spontaneous. It happens without activation energy of its own. It happens by itself. And it's increasing what else? Entropy. It's causing disorder to increase in the universe. So negative delta G, it's releasing energy. It's exergonic, spontaneous, and it increases entropy. Positive delta G is just the opposite. So make this be a positive number. Positive delta G means it's doing what with energy? It's absorbing it, it takes it in, it has to have it in order to get started, right? So it's what kind of reaction? Endergonic, it takes in energy. It's not spontaneous. It has to have that energy to get started. And what's it doing with entropy, at least in a lot of cases? Decreasing it. Because a lot of times these are anabolic kinds of reactions, right? They're building up things. One other thing on this slide that I want you to know, the breakdown of ATP, the hydrolysis of ATP, is obviously an exergonic reaction. It releases about negative 13 joules of energy per mole. On the other hand, the making of ATP, the synthesis of ATP, takes in about 13 joules per mole. So those are just two examples of delta Gs. You probably ought to know that number 13. Releasing ATP gives off 13 joules. Making ATP takes in 13 joules. Yeah.
You said one thing where it's spontaneous, it's exergonic. Or spontaneous, not spontaneous, for endergonic or exergonic. Increase or decrease in entropy.
And does it take in energy or give off energy?
Wait.
Take in energy, give off energy.
So.
Which one are you asking me about?
Well, I'm looking at the negative one, but.
Okay, a negative delta G means that it gives off energy. It's exergonic. Those mean the same thing. They really mean the same thing. Exergonic means it gives off energy.
Okay, that's why I'm confused.
Yes, mean the same thing.
Okay.
It's spontaneous, it increases entropy. Everybody okay here on this slide? Be able to explain that. That will show up in multiple ways on your next quiz. Here's another idea about coupling. This reaction down here at the bottom must be what kind of reaction? Endergonic. Because we know ATP hydrolysis, which is what that is what kind of reaction? It's exergonic. This one, the exergonic reaction, provides the activation energy or the free energy to make that one happen, right? They're coupled. This one up top, ATP hydrolysis, provides the activation or free energy needed to make the endergonic reaction at the bottom happen. Does that make sense? This is the way your body works. This is like having gas in your tank. The gas makes the engine run. Same kind of thing. All right, most of what we're going to hit today is some serious stuff about enzymes. When you hear the word enzyme, first thing I want you to think about is what they're made out of, which is what? Proteins. Which means they're coded for directly by what? DNA, genes. If you have a mutated gene for a specific enzyme, then that enzyme is not going to have the right way. If the gene for a particular enzyme is mutated, the enzyme is going to end up with the wrong way. Wrong shape. The active site is going to be shaped the wrong way, which means it's not going to work. It's not going to work. Which means there's a chemical reaction that you need to do, but now you can't do, right? That can be really serious. That causes all kinds of genetic diseases. When we get into genetics, we'll talk about Tay-Sachs disease. Talk about another disease called PKU. And these are all diseases caused by defective enzymes. Enzymes that are made the wrong way because of a mutation. Other couple of things I want you to think about enzymes is they're catalysts, which means they help to get reactions started. They help to speed them up, but they're not actually what? Yeah, they don't actually take part in the reaction. They help to get it started, but they don't actually take part in it. They're not changed by it, which means what about an enzyme? It can be used over and over and over and over again. It's not used up. Another thing about them, and a really important idea, is they decrease the amount of free energy or activation energy that you need to make a reaction happen. They decrease the amount of energy needed to make a reaction happen, the free energy or activation energy. And we'll talk about how that works here in just a second. Now, we've talked over and over again about protein structure. Primary, secondary, tertiary, quaternary. You've got to know those things. You need to know what they are, the bind to hold them together, all that stuff. I'm not going to go through that again because we've been through it about 5 or 6 times. Proteins, though, enzymes are proteins. Shape is important. Before I go further, I want to look at a picture. This is supposed to be an enzyme. An enzyme is a big 3-dimensional globular protein. It's like a big glob of protein. So if that's the case, what would each one of those little blue spheres represent? Tell me out. Each of one of those little blue spheres in there, what do they represent? That's an enzyme. It is a protein. So what is each sphere? It's an amino acid. There's a bunch of amino acids all hooked together, wrapped together in this specific shape. Remember that probably the most important part of an enzyme is this little groove right here, which we call the active site. What are the 2 things that are important about that groove? What 2 qualities are the most important about that groove? Its shape and its charge. The shape and the charge of that groove have to be complementary to what? The shape and charge of the substrate. That's where they get together. That's where they fit together. What is the substrate? What is a substrate? Yeah, it's the thing or things that an enzyme works on, acts upon. For example, what's the enzyme in saliva? Amylase. That's the enzyme. What's its substrate? What's amylase work on? Starch. We're going to work with an enzyme in the lab on Monday called catalase. We've mentioned that in here before. What's catalase's substrate?
Hydrogen peroxide.
Hydrogen peroxide. What's the breakdown peroxide? What does it break peroxide down into? Water and oxygen gas. And that's going to be important to know for the lab, by the way. But here's the thing. The enzyme and the substrate, the active site rather, and the substrate have to be really complementary in terms of shape and charge. They have to fit. And we say they fit like what? Like a lock and key. What does that mean about each enzyme? Yeah, it can only fit one specific kind of substrate. It can only catalyze one specific kind of reaction. Enzymes are specific. There is no jack of all trades enzymes that can do all kinds of stuff. Each enzyme does one thing. So what does that mean about the number of enzymes you have in your body? You got a bunch of different ones. A whole bunch. Each one does a different thing. Now another important point to talk about. You see these 2 arrows here. The substrate fit in, and then what did the enzyme do? It had a slight change in shape, sort of closed around it. Let's say that this enzyme on the screen is an enzyme that's doing anabolic work. It's putting pieces together. And let's just use my hands as an analogy here. Let's say that my hand here is the active site, and these are the substrates. And they fit in. They fit in the groove. And then once they fit in, the enzyme is triggered to what? What's going on up there? Move a little bit. And which way are these going to move if it's an anabolic enzyme, probably? Like that, right? So when it squeezes in, it helps to push 2 substrates together. That slight change in shape of an enzyme that helps to make it work is called what? The arrows represent what's called an induced fit. That's an important idea to note. Induced fit. It's just one T there, by the way. Induced fit. It's a slight change in shape that helps the enzyme to work. It improves the way the substrate fits in the active site. If on the other hand, this enzyme, my hand in this case, was a catabolic kind of enzyme, substrate fits in there, probably what's the enzyme going to do? The catabolic enzyme. Once it fits in there, what's it going to do? It's going to pull this way, so it breaks the line. It's going to put stress on those lines. But an induced fit is when the shape of an enzyme slightly changes to make it work more effectively. That's very different than what other change in shape of protein we talked about. Denaturation, very different. This is a good thing. This makes the enzyme work. Denaturation basically makes it what? Fall apart, not work. Two different things. All right, I'm going to back up a little bit. All right, I think I'll look at this, remind you about this picture right here first. Most people hear the word enzyme and they think about digestive enzymes. They think about breakdown, catabolic, hydrolysis. But as you can see in this diagram, there are enzymes that do both catabolism and anabolic kinds of reactions. Here we've got synthesis going on. Dehydration, condensation, anabolic kinds of things. Notice that these substrates have the shape and charge that are complementary to the shape and charge of that active site. They fit in. So we say enzyme plus substrate. When they fit together, we call that the enzyme-substrate complex. That's when what's the enzyme going to do when they both fit together in there? What we say it's called when the enzyme slightly changes shape. Induced fit. So that's going on in here. And those two substrates are being lined up and reacted together, joined, synthesized. And what we'll end up with is the enzyme. It separates, it's unchanged, and we'll get a product or products. Same kind of thing happens over here, except this is more of a catabolic kind of thing, a degradation or hydrolysis kind of thing going on. The substrate again is shaped so that it fits the groove, the active site. It fits in. Here we get that induced fit going on. In this case, it's probably going to bend so that the bind between the two pieces of the substrate get broken. We still get the enzyme, just like it was at the beginning, unchanged, and then we get our products. Does that make sense? All right, now we've said over and over again that enzymes work by lowering the amount of free energy or the amount of activation energy needed to start up a reaction. Well, here's 3 ways that they do that. Before you try to write any of that down, listen to me for just a second. Let me explain something. Let's say that this little thing right here is an active site. It's an enzyme. That's the groove's the active site. And this thing right here is the substrate. They go forward. Right, if they're just floating around in a container of water, how likely are they to find each other? Not very, right? What can we do to the container of water to increase the likelihood that they would find each other? We could heat the water up. Because if we heat it up, what's that going to change about these things? They're going to move faster. And they're more likely to collide into each other, right? But if they collide like this, is anything going to happen? If they collide like that, is anything going to happen? Like that, nothing's going to happen. Like this, nothing's going to happen. They've got to collide the right way for something to happen, right? They've got to collide like that. Kind of just chance. So we could heat the water up. What else could we do to increase the likelihood that they would find each other? Yeah, we could add more to the substrate, increase the concentration of the substrate, or we could also increase the concentration of the enzyme. But let's go back to another idea here for a second. Let's switch gears. Let's say these two markers represent substrates, and they're floating around in a container of water. Right now, we got no enzyme in there. It's just the substrates. These are two things that will react if they collide the right way and they line up. But they're just inside a container of water right now, floating around. What determines if they're going to collide the right way and react or not? There's no enzyme in there. What determines whether or not they collide and react? They're just floating. Why are they moving in the first place? What do we call that random motion of things because of heat? Yeah, there's another word for it though. Just vibrations. What kind of motion? Brownian motion. Remember that term? Brownian motion. But the thing is, if they collide like this, is anything going to happen? Nope. They collide like that. Nothing's going to happen. They got to collide like that. It's just random if there's no enzyme in there, right? Maybe they will, maybe they won't. One of the things that enzymes do is an enzyme might encounter this particular substrate. It'll grab it. It'll hold it in its active site in a certain orientation. And then as it's moving around, it'll come in contact with the other one. It'll grab it. And what does it do to those two substrates when they're in the active site? What's the enzyme done to them? It lined them up. It put them in the right orientation so that when it does that little induced fit thing, what's going to happen? It's going to push them together. That's all number one to say, is that one of the ways that enzymes lower activation energy is they line up the substrates so that they'll react. So that the substrates aren't dependent on just randomness to line up. The enzyme lines them up so they're more likely to react. Another thing they do is I sort of just showed you a second ago. Let's say this substrate, my two markers here, fits into the active site. What did we say the enzyme is going to do once it fits in there? If it's a catabolic enzyme, what's it going to do? You get the induced fit thing going on. So it pulls, and it starts to put some stress on that bond in there. And eventually, if it puts enough stress, it's going to break, right? That's all we're saying, is when the substrate's bonded to the active site, the bonds within that molecule might get stressed because of the induced fit of the enzyme, and that causes it to break. So this kind of thing would apply to what kinds of enzymes? The first one, substrate orientation. What kind of enzymes are doing that? Anabolic or catabolic? Anabolic. This one would apply to catabolic, right? Breaking. Now the third way enzymes can change the activation energy, lower it, is to do what's called changes in substrate reactivity. They can add electrons to a substrate or take electrons away from a substrate. Electrons typically carry what with them? If I was to give Alex some electrons right now, what else am I giving Alex? True. Negative charge. And look at those lights working up there right now. Movement of electrons. Electricity. So what are electrons carrying with them? They can make a light light up, what must they be carrying with them? Energy. So you can transfer energy by transferring electrons. Now in chemistry, you should have heard of two terms that are going to be really important. Actually, sometimes it's one term all smushed together. Those kinds of reactions in which electrons are moved from one thing to another are called redox reactions. R-E-D-O-X. Important word to know, redox. What does it stand for? Redox. Stands for reduction and what? You guys shouldn't get that information. Reduction and oxidation. Now, you need to know some definitions of those two terms, because it's going to be real important. If we oxidize something, oxidation, what do you think that means? It seems like it ought to mean you react it with oxygen, right? And that was the original meaning. But what they figured out, what do we know about oxygen? What have we said about oxygen over and over again in this class? What does oxygen like to do? It's got a high electronegativity, right? It wants to pull electrons away from things. Turns out when oxidation, the meaning of oxidation now is when you take electrons away from something. Because that's what oxygen likes to do, right? It's not the only thing that likes to do it, though. Oxidation is when electrons are removed from something. Which means, what else is removed from that something? Take away electrons. What do we say electrons bring with them? Energy. If you oxidize something, you're taking away electrons, taking away energy. Reduction is just the opposite. If you're reducing something, you're adding electrons to it, therefore adding energy to it. Why is it called reduction then? That doesn't make sense. If you're adding to something, why is it called reduction? Let's take an example. Let's say that we have an oxygen atom. How many electrons does an oxygen atom have? Come around and look at the periodic table.
What?
How many electrons? Not valence electrons. How many total electrons does an oxygen atom have? 8. Atomic number should give you the number of what? Protons. And if it's a neutral atom, it should also be the number of electrons. So it starts off with 8 positive protons and 8 negative electrons. Well, let's say we reduce that oxygen atom. Let's say we give it 2 extra electrons. So now instead of this being 8, now it's got 10 negatives. What did we reduce about that oxygen? Before, it had 8 positives and 8 negatives, which meant its charge was what? 0. After we reduced it, now it's got 8 positives, but 10 negatives. So what's its charge? -2. What went down? The charge. That's why it's called reduction. When you add electrons to stuff, the charge goes down. Does that make sense? All right. Let's try to finish this up. Let's see. Let's talk about this little phrase right here. Read that phrase. Kind of makes you sound smart when you say that, right? Complementarity leads to a high degree of specificity. You sound really smart. You go around and say that and people think you're a genius. Complementarity leads to a high degree of specificity. What does that mean in terms of enzymes and substrates and such? What do we say has to be complementary? The shape and charge of the active site have to be complementary to the shape and charge of the substrate. That's the complementarity part. And because of that, what does that mean about every enzyme? They have a very, very specific thing they do. Can't do more than one thing. Each enzyme is specific. It catalyzes one reaction, period. So that really sounds complicated, but it's really not so complicated. It's pretty simple. The shape and charge of the active sites have to be complementary to the shape and charge of the substrate. And because of that, each enzyme is highly specific. Only does one thing. Another kind of cool thing to think about, most enzymes in your body are found in only certain places. But we know that enzymes are proteins. And we know proteins are coded for by DNA. What do we know about the DNA in every cell in your body? We took a cell from Matt's scalp, and we took a cell from Matt's liver, and we compared the DNA. What should we find? It's exactly the same. But are the proteins in Matt's scalp the same as the proteins in Matt's liver? Nope. Why not? Genes get turned on and off in different places in your body. The cells in Matt's scalp are making very different proteins than the ones in his liver, even though they have the same ability to make the same ones. Think of your genes like light switches. They can be turned on and off. And they get turned on and off based on signals from your body. The signals that are saying in your stomach to turn on certain kinds of digestive enzymes. You don't want those digestive enzymes to be turned on in, say, your nose, right? Because otherwise your nose would disappear. You would digest your own nose. That would be bad. You don't want digestive enzymes in the wrong parts of your body. Your body can control by turning on and off certain genes where certain enzymes are found. Not that I know of, but it's possible. Could happen. All right, the next thing we're going to talk about is kind of interesting. You've all been told that you need your vitamins and your minerals, right? How many of you take like a vitamin tablet every day? Not a bad idea. You've probably been taking them and didn't really know why you needed them. I'm going to tell you, you take vitamins and minerals because they help your enzymes to work. Period. That's the reason we need vitamins and minerals. No. Minerals. Let's talk about minerals first of all. What is a mineral? What is a mineral? It's usually a metal ion that your body needs. Metal ion. Minerals are metals. When I say you need iron in your body, am I talking about iron? Yeah. When I say you need copper in your body, I'm talking about the same stuff that the top of the penny is made out of.
Yes.
Zinc. Yes. The same stuff you played with in chemistry. You need that in your body. Magnesium. The same stuff that you lit on fire in Chem 1 class and it glowed like a flare. You need that in your body. Turns out magnesium is the element that in a plant, what is built around. It's the most important protein in the plant. The most important protein in the plant. This is not a difficult question. What is the most important protein in the plant? The plant, guys. It's green. What is the most important protein in the plant? Chlorophyll. Okay? Plants need magnesium to build chlorophyll. You need iron to build what? What important protein in you is built around iron? You all should know this. What protein in you is built around iron? What happens if you don't have enough iron? You ladies should know this specifically. You feel real tired. It's more of a woman problem than a guy problem. Why?
Because we lose our third.
Okay, so where's iron at in your body?
And our siblings.
Blood. What's the major protein in your blood? Tell me out here. Don't make it hard. Hemoglobin.
I said that.
I didn't hear you, I'm sorry. Hemoglobin's job is to carry what? No. It's in your blood. What is the major job of your blood? To carry oxygen. So that your cells get oxygen so they can do cellular respiration. If you don't have enough iron, that means your body can't transport enough oxygen, which means you can't make enough what? ATP, which means you're going to feel tired and yucky. Okay? And I said it's more of a woman problem than a guy problem, low iron, because you lose blood during your menstrual cycle. And because us guys tend to eat a whole lot of what? Red meat, which has a lot of what in it? Iron. Okay? So us guys typically don't get iron deficiencies. You ladies sometimes do. What do we call an iron deficiency?
Oh, first of the day.
Anemia.
Anemia.
Yeah.
Okay? Anemia.
Sorry.
It's okay.
That's okay. The key thing,
Get that one.
minerals. Minerals are metal ions that certain specific enzymes need to work. They actually bind to the enzyme, and if the enzyme doesn't have them, it won't work. We call minerals that your enzymes need cofactors. So minerals that bind to enzymes and make them work, they're called cofactors. Now a lot of people get minerals confused with vitamins because their functions are similar. What they're made out of is very different. Vitamins are big organic molecules. They're made out of what? Big organic molecules are made out of carbon. Okay, they're carbon. They're not metals. They're carbon. Carbon-based things. Some hydrogens. But their function still is they attach to enzymes and they activate the enzymes. They make them work. So their function is essentially the same as minerals. If it's a vitamin that attaches to an enzyme and makes it work, we call it a coenzyme. If it's a mineral that attaches to an enzyme and makes it work, we call it a cofactor. So minerals are cofactors, vitamins are coenzymes. But the net result is this. Both of those things bind to a specific part on a certain enzyme and make it work. They activate it. Let's talk about some problems when you don't have the right vitamins. Let's say you're a woman who's pregnant many years from now. And you don't have a specific vitamin known as folic acid. You've got a deficiency of folic acid. You probably heard of that on orange juice commercials. They talk about it on orange juice commercials. Folic acid. And turns out folic acid is a coenzyme for the enzymes that help to build your nervous system. So the enzymes that build the nervous system won't work without that. Children born to women with this folic acid deficiency oftentimes end up with a condition called anencephaly. Let's break that word apart for a minute. This is kind of a scary thing. What's an mean? No or not. What does cephalo mean? Or cephaly? Like in biology, you probably talk about cephalopods, like the octopus and the squid. No? Cephalo means head. This literally means no head. Now, well, they have a head, but what they don't have inside their head is the main part of their brain.
Oh.
What is the main part of the human brain called? The big part that makes us think and remember and have feelings and memories and consciously do stuff. Well, it's parts that's divided into the lobes. But when you see a picture of a brain, this is really the only part you probably pay attention to. It's the huge part of the brain. It's called the cerebrum. These children are born without a cerebrum, but they're born alive. The cerebrum is the part of the brain that makes you think, makes you remember, makes you be conscious, know you exist. It's the part that helps you feel. It's the part that lets you consciously control things.
So these babies are sick.
Yes. Here's where I'm going. The back part of your brain, the part that's connected to the spinal cord, is called the brainstem. And you've heard of one part of that before. What's the famous part of the brainstem that you've all heard of? With a really cool sounding name. It's called the medulla oblongata. You've all heard of that before, right? Medulla oblongata. These babies have that. They have medulla. The medulla's job is to make the heartbeat. to make the lungs work. So they're alive, but they'll never wake up. They don't have the part of the brain that makes you be conscious, aware, awake. They can't move on their own. They can't feel anything. They can't. They may live for weeks or months, but they'll never do anything on their own. They don't have the ability to do that. So they have to put in a feeding tube if they decide to try to keep them alive. But if you keep them alive, they're still never going to know they're alive. So it's strange. The moral of that story, it's not that uncommon. I don't know the statistics.
So, like, sick women get a lot of folic acid.
Yeah, if you're planning on having a child, you should always take prenatal vitamins before you get pregnant, right? And throughout the pregnancy, take the vitamins. Say again? Well, folic acid, I think, is a water-soluble item, and you just urinate it out if you have too much. So that's not a problem. Take your vitamins if you're going to have a baby. Let's talk about another example of this. Vitamin C. What do we call the disease you get if you don't have enough vitamin C? Scurvy. Who used to get scurvy? People that were ship people in general. Because typically in our diet, where do we get vitamin C from? Fruits, like citrusy fruits, right? If you were sailing from Spain to North America, can you keep citrus fruits for that long? No. So they weren't getting any vitamin C. What are symptoms of not having vitamin C? Your gums start to bleed real bad, teeth start to fall out. And then it gets worse, because it turns out that vitamin C, is a coenzyme. Vitamin C is a coenzyme for the enzymes in your body that build collagen. What's collagen?
So that makes your bones.
It's the strongest protein in your body. It's the protein that holds you together. It makes up your connective tissues. So if you don't have vitamin C, you literally start falling apart because you can't make that. And prolonged scurvy can cause blood vessels in your body to start to burst because collagen makes up the walls of those blood vessels. What do we call it when a blood vessel bursts inside your body? Aneurysms. It causes internal bleeding. So that can kill you. And it's simply because you don't have vitamin C, which is a coenzyme for the enzymes that make that protein. The enzymes that make that protein won't work without vitamin C.
I partly does not eat anything healthy like vitamins or like fruits or anything like that. So you need to tell that you talk with her again and know that she's gonna fall apart and her lips or her gums are gonna bleed.
Water and food like a nutrient, like that kind of healthy stuff, but nothing like no vegetables.
Right. Or fruit.
Nothing real.
I'll make sure for
I
yeah.
have an internal bleeding question.
Yes.
Don't laugh. I will. I get that you're bleeding on the inside, but where does it go?
It just pulls up.
Well, where? Like underneath
In your cavity. I mean, you got a lot of empty space in here, right? There's places it can just pool up, literally. I mean, if we were to cut you open, there's a lot of organs in there, but there's also just a lot of space. Well, you a lot of times you won't. Or I mean, internal bleeding can also be like when your stomach is bleeding inside the stomach. So when you go to the bathroom, you can see it, the blood. I mean, there's blood in it. Or you can have internal bleeding in, say, your kidney or your bladder, and when you urinated, you would see that blood. So there's lots of different ways that can happen.
So, never mind. I didn't really have a question that time. Yeah, okay.
We got it now.
Yeah.
Let's talk about another vitamin deficiency. Let's say you don't get enough vitamin A. Where in your diet do you normally get vitamin A? orangey, yellow kinds of stuff, like carrots, sweet potatoes, stuff like that. Vitamin A is a coenzyme for enzymes that make sight work. You've heard that carrots are good for your eyes. That's true. Okay? It's true. Inside the heart of Africa, where people are really, really poor, how many carrots do you think they have to eat? Probably none. So they're not eating a lot of orangey, yellow things. What are these folks eating? Really, really poor people in Africa.
And
Yeah.
beans.
Rice,
and
grain things.
corn.
A lot of times they call it gruel. It's just this rice, like soupy stuff. Now, rice is really good for energy. But how much nutritional value does it have? Not very much. So these people, that's about all they get to eat. They're not getting vitamins and minerals. If they have it for a prolonged period of time, they get what's called night blindness, which means when it starts getting dark, you can't see anything. And if it's prolonged vitamin A deficiency, they go blind, not just not blind, but blind. Because the enzymes that make sight work can't work without vitamin A, so it's coenzyme. Vitamin K, that's another important. Vitamin K, an adult gets, the bacteria that live in your intestines actually manufacture it for you. Anybody know what you need vitamin K for? First of all, it has nothing to do with potassium. Completely different thing. Nothing to do with potassium. Vitamin K. A lot of people have asked me about it. Nothing to do with potassium. Vitamin K. It's a coenzyme for enzymes that make your blood clot. And your blood won't clot without vitamin K. I'm going to tell you though that I said that we typically get it, adults, and people your age too, from bacteria that live in your intestines. They make it for you. A newborn baby, do they have bacteria in their intestines? No. One of the first things that the hospitals do to newborn babies in this country is they give them a shot of vitamin K. Coming out of the mother, it's kind of rough on the baby, right? They can get injuries, like coming out of the mother. Like some babies are born with like shoulders that are out of socket, and their heads are shaped like a cone because they got squeezed on the way out. Yeah. So point being, all these injuries without vitamin K, what could happen? What can happen if your head is squeezed and you don't have any vitamin K? You can bleed to death inside. Internal bleeding. There we go. Right? So they give them vitamin K injections in parts of the world where they don't do that. Some babies bleed to death. Where do you get those bacteria at that are in your intestines? How do you get them? You get them from eating. Well, those babies have never eaten before, right? You get them from milk, and you get them from breast milk, but it takes a while to build them up and to get enough vitamin K. One more vitamin I'll mention is vitamin D. What's vitamin D involved in? It's involved in coenzyme for enzymes that build your bone. If you don't have enough vitamin D, you get a condition called rickets. Your bones are really fragile and weak, and they'll break just from like standing up, kind of stuff. Okay.
Okay, old people with like, you know, the like humps. How does that happen?
Like an actual hump on their back, or their back
Yeah,
is
like
just
they're
being.
hunched over.
There's several things that can cause it. Osteoporosis can be a weakening of the bones. That can cause it. That's mostly the main. Another thing is you can have a disorder called what's the name of it? Graves' disease. It's a problem with your thyroid gland, and it can cause you to swell, and it can cause you what's called buffalo hump on your back. So there's several things that can cause it.
I didn't remember what it was.
We'll get you when we come back to it. So key thing, vitamins and minerals, you need them so that your enzymes will work properly. If you don't have them, deficiencies cause certain enzymes in your body not to work. Yes?
I got it. Do you have to have calcium like for your bones? Like if you don't drink calcium, are your bones being weak?
Yes.
Because I want to drink milk.
Well, but
You can get calcium from other
You
things.
can get calcium from other places. You don't have to drink milk. I mean, cheese, yogurt, ice cream,
Ice cream.
certain vegetables. You can get calcium in a lot of places.
Mom, I gotta eat my ice cream today.
There you go. It's good for your. Tell her that I said you needed some ice cream to be healthy. All right, let's talk about these two graphs. This is what we're going to be doing in the lab on Monday. You're going to end up having to make very similar graphs. This is looking at the rate of a reaction, a rate of an enzyme catalyzed reaction compared to temperature. And what you notice is as you heat up the reaction, what's happening to the rate? It's going up. It goes up to here. That's the temperature it works best at, right? What's a word for best? Optimum. That's the optimum temperature, the temperature it works best at. In your body, what's the optimum temperature for just about every reaction that happens in your body? 98.6 Fahrenheit, which is what in Celsius? It's the number you need to know. What does that look like on that scale up there? Is the optimum temperature? 37. 37 Celsius is about the same as 98.6 Fahrenheit. 37-ish. So if we went straight up to here, that's somewhere around 37. Somewhere around 37. Now, here's the deal. Why does heating up a reaction at first help it to speed up? Heat makes things move faster, which means it's easier for what to find what? If they're moving faster, it's easier for substrate to find the active site. On the other hand, once we pass the optimum temperature and get hotter and hotter, these things are still moving faster and faster and faster. But why does the rate of the reaction start to drop past that optimum temperature? What happens to this enzyme past the optimum? We call that denaturation, and it falls apart, right? All those hydrogen bonds and disulfide bonds break, it starts to fall into pieces. Here's another kind of interesting idea. What's a reaction in your body that happens best at temperatures less than 37 degrees Celsius? You heard this before, that's why you're dancing. Turns out this is only in some of you. Think about this. Nature, God, however you want to think about it, spent a lot of effort to make guys bigger and stronger than women. And then there's just one obvious weak spot, correct? And it doesn't make sense, right? But that's where it's at. Turns out it has to be there because the enzymes that make sperm work best when? Under what conditions? Temperatures less than 37. And the only way to get the temperature less than 37 is to get those parts where? Outside of the main core of the body. It's cooler there. And it's the only way the sperm can be made properly. The enzymes that make them work at temperatures less than 37. You've heard this thing about guys wearing tidy bodies and that makes them sterile? Heard that before? Might there be something to that?
They're all.
Yeah, you don't have to draw.
Get all hot and stuff. And then,
Yeah.
yeah.
Those parts get pushed closer to the core of the body and therefore get warmer and therefore don't work as well. You might have heard the idea that guys that use laptop computers on their laptops are more likely to be sterile. Probably something to that. But laptops give off a lot of heat, right? Increased temperatures. Does that make sense?
We shouldn't children.
Oh,
Well, the children probably aren't trying to have children, so it's probably
I expect them sooner or later.
that's probably a short-term kind of problem, so probably. Probably.
Waiting for it, so we should order that anyway.
No, I go in there. Let's look at this other graph. Let's move away from that. We're looking at rate of reaction against pH. What is the optimum pH for just about every reaction in your body? 7.35 is the magic number, right? That's the pH of your blood and your body fluids. Just about every reaction in your body works best at 7.35. This one's pretty close to that, right? This one over here is not. What's the optimum for that particular enzyme? 2. Where's that enzyme probably found at in your body? Where in your body might the pH be 2? In the stomach. Stomach acid, right? This turns out pepsin is an enzyme found in your stomach that helps you to digest proteins. That's one of the only places in your body where the pH shouldn't be 7.35. Your stomach. But notice that if you change the pH, either make it more acidic or more basic, what starts to happen to the rate of the reaction for either one of those? If you move the pH away from the optimum, what happens to the rate? It drops, because what starts happening to the enzyme starts to fall apart and denature.
Christina, I have a question about
Yeah, PEP usually refers to what? Well, it usually refers to proteins like peptides, polypeptides. But we said before about the only thing that happens in your stomach to your food is what? The digestion of proteins. So sometimes we use pep to mean stomach as well. Like if an ulcer is called a peptic ulcer, it's in your stomach. Pepto-Bismol is supposed to make your stomach feel better. It's a bacterial infection, treatable with antibiotics. They didn't know that for many, many years.
Does it hurt?
Can, but the more dangerous thing is it can eat into your stomach wall, and you can start to have Maddie's favorite word, internal bleeding. And it can actually, you can bleed to death from an ulcer. It can be really dangerous. You're going to say something, Paige?
Yeah. Like if you take like 2, maybe to like do something.
There are certain drugs that might change the pH of your stomach and might cause your acid to start eating through the walls. It's possible. All right, the last topic I want to talk about today, and we'll mention the lab a little bit, are inhibitors. This is kind of a cool topic too. What does inhibit mean? Stop. You inhibit something, you stop it. Inhibitors are things that stop enzymes from working. Period. Inhibitors are things that stop enzymes from working. We're going to talk about 2 main kinds of inhibitors: competitive and non-competitive. Competitive inhibitors compete with what for what? What do you think they're competing with? They're competing with the substrate for the active site. Competitive inhibitors compete with the substrate for the active site. Which means, so what does that mean about the shape of that competitive inhibitor probably? If it can fit into the active site, that means what about its shape? It must be really similar to the substrate. Its shape and charge must be real similar to the substrate. Because we said they fit together like what? Like in a key. So the shape's got to be similar. But if the inhibitor is bound to the active site, what can't get there? The substrate. So the enzyme is effectively not working. Off. It's off. Let's talk about an example of a competitive inhibitor. Anybody in here allergic to penicillin? A lot of times if you're allergic to penicillin, instead of giving you a normal antibiotic, they'll give you this category of drugs called a sulfa drug. They kill bacteria, but they're made a little bit differently. They're different chemical than penicillin and amoxicillin and those other antibiotics. Here's how they work. Folic acid we talked about a minute ago. We said that's a vitamin that we need, right? But where do we get our folic acid from? humans get folic acid from where? From our diet, orange juice and other fruits and vegetables. Bacteria also have to have folic acid. But they don't get it from their diet. They have enzymes that help them to make it. That's the key point. Bacteria make their own folic acid. We eat or drink ours. These sulfa drugs work by going in and blocking the enzymes, inhibiting the enzymes that help the bacteria to make folic acid. If bacteria don't have folic acid, what happens? They die. So these drugs block the enzymes in bacteria that make this vitamin for them, folic acid. These drugs don't make you sick because you don't have what? You don't have enzymes to make folic acid. You get your folic acid from where? You eat it. You don't make it. So you kill the bacteria by inhibiting an enzyme that they have that you don't. Turns out that's how most antibiotics work. They inhibit enzymes that bacteria have that we don't. So therefore, it kills the bacteria. Doesn't hurt us. Let's talk about non-competitive inhibitors for a minute. Maybe the most famous one of these is penicillin. How long have we had penicillin? Probably the most important discovery in science ever was the discovery of penicillin. When did that happen? Around the 40s. It was discovered a little bit earlier, but we didn't actually figure out how to use it until somewhere around the end of World War II. First people that penicillin was used on were soldiers from World War II, American soldiers and British soldiers from World War II. When they discovered penicillin, they thought they had cured what? What's penicillin used to treat? What kind of diseases? Bacterial diseases, not diabetes or cancer. No, not those.
What is it, diabetes? What is that?
Diabetes is a cancer, is a problem of the pancreas. Use insulin. Right. Use insulin. But the key thing is they thought they had cured bacterial diseases. Now we know today it didn't quite work that way, right? Because what did bacteria do fairly quickly? They mutated and became resistant to penicillin. A lot of things that penicillin used to kill, it doesn't anymore. But here's the way penicillin works. What structure does a bacterial cell have that a human cell does not? A wall. Turns out that penicillin works by inhibiting enzymes that build cell walls. So it keeps the bacteria from being able to what? They can't build a wall, so what can they not do? Well, but they can't make new bacteria. They can't reproduce. How long does a typical bacterial cell live? A few hours. But if they can't reproduce, what happens to the bacteria real fast? They go away. Penicillin doesn't make you sick because you don't have what? Enzymes to build cell walls. We inhibit an enzyme in bacteria that we don't have. That's how most antibiotics work. Yeah.
Sulfa drugs, if you can take what?
Penicillin. Now let's talk about some other things that are inhibitors. Heavy metals. When I use that word heavy metal, what am I talking about? Yeah, I made that joke yesterday. Talking about Van Halen and ACDC, right? Yeah. What are we talking about when we talk about heavy metals? Mercury, lead. Those are the main ones. Cadmium. If Joanna here was to pick up a bottle of mercury and drink it, she would very quickly not be with us Monday, all right? Mercury. Okay? Mercury binds to certain enzymes in your body and keeps them from working. Specifically, the most important places that it binds are enzymes in your brain. It causes you to go crazy and eventually just to stop working. Lead does similar things, right? Lead poisoning.
They used to put mercury and lead in hats, right?
Yes, that's what Mad Hatter in Alice in Wonderland was a loony because they used to use mercury in the process of making hats. This was going on when Lewis Carroll wrote Alice in Wonderland. And they didn't really know why they were going crazy. Eventually figured out it was mercury. They just thought hat makers were crazy, I guess. But it was because of the mercury. Lead poisoning. That's similar kinds of things to you. Makes your brain not work properly. And given enough ingestion of lead, it will kill you. There are some people that think the Roman Empire fell because the Romans were using what? Lead to make pipes and plates and cups and all kinds of stuff. And then every time they drank something, they were drinking lead. And it, like the Roman emperors, who's like the famous crazy Roman emperors? Nero did all kinds of crazy stuff. Caligula was another one. You might not have heard of Caligula. But Caligula made his horse a senator. His horse actually went to Senate meetings. I don't know how he voted. I think he always voted no, right? Nay. But the point being, that was pretty good. Point being, heavy metals are dangerous because they're enzyme inhibitors. They keep certain things from working in your body. Most poisons are dangerous because they stop specific enzymes from working. Rattlesnake venom stops certain enzymes from working in your body. Arsenic, cyanide, common poisons work because they keep certain enzymes from working in your body. Even chemical weapons, like mustard gas, those work because they inhibit very specific enzymes in your body. And if those enzymes don't work, you die. So enzyme inhibitors are serious business. If certain enzymes in your body don't work, you won't last very long. And they can be either competitive or non-competitive. Either way, they're dangerous. All right, I'll stop there. Let me mention two quick things before we go. All right.